The effects of climate change and rising global temperatures will continue to pose an existential threat to the agriculture industry for decades to come. Increases in biotic stressors could adversely affect our food supply by significantly reducing the yield of valuable crop plants. For example, threats such as insect herbivores will continue to increase in population size, geographic range, and feeding voracity as global temperatures continue to rise. Now, the KED gene was designated this name based on the amino acid composition of the protein that this gene translates, which is rich in lysine, glutamic acid, and aspartic acid. Previous studies concluded that this gene is indeed involved in the wound-induced mechanism of tomato plants, but the implication that this gene is involved in the defense system against biotic stress, such as insect herbivory, remains inconclusive. Previous studies have generated a line of tomato plants with a non-functional KED, and they did this by using the CRISPR-Cas9 system to knock out the KED gene. Our lab has performed insect assays using the tobacco hornworm that was fed on both wild-type and KED knockout samples. Now, our results displayed that hornworms that were fed on the KED knockout samples not only gained more mass and grew longer than those that were fed on wild-type samples, but they did so at a faster rate. Now, in order to further investigate the role of the KED in the defense system against biotic stress, there are a couple experiments we must work on. So we must further analyze the release of defense-induced secondary metabolites. We must analyze gene expression under variable conditions, and we must analyze the effect of the KED gene on feeding behavior and metabolic activity of the tobacco hornworm. To conclude, we hope that this research proves to be a valuable asset to not only the agriculture industry, but to society as a whole, as it could provide to protect our food crop in times of increased stress, such as those magnified by climate change.